Welcome to this lecture on developmental biology. Today, we are going to explore one of the most fascinating and vital structures in mammalian reproduction, the placenta. Imagine an astronaut floating in the dark, cold vacuum of space. To survive, they rely entirely on a tethered life support backpack that pumps in fresh oxygen, delivers liquid nutrients, and pulls away toxic carbon dioxide. For a developing baby inside the womb, the environment is just as isolated, and they need their own ultimate life support system. In this video, we will uncover how the placenta forms, the different types that exist across species, and the mathematical principles that govern how it keeps a fetus alive. Let us dive in. Building on that astronaut analogy, let us consider the physical reality of the fetus. While submerged in amniotic fluid, the developing baby is completely isolated from the outside world. It cannot breathe air, it cannot digest food, and it cannot urinate on its own. Therefore, it relies entirely on a biological life support system to perform all of these critical survival functions. The placenta acts as the baby's lungs, digestive system, and kidneys all rolled into one temporary, miraculous organ. Without this vital connection, the tiny developing life would have absolutely no way to acquire the building blocks it needs to grow, nor could it get rid of the toxic metabolic waste it constantly produces. Now that we understand why the fetus needs this support, let us look at what the placenta actually is. In developmental biology, the placenta is defined as a temporary, specialized organ. It is unique because it is formed by the fusion of tissues from two entirely different individuals. The fetal contribution comes from the extraembryonic membranes, specifically a layer called the chorion. This fetal tissue fuses intimately with the maternal contribution, which is the uterine lining, known as the endometrium. Together, they create a highly vascularized interface. This dual origin is what allows the placenta to act as a perfect bridge, facilitating communication and exchange between the mother's body and the developing baby throughout the entire pregnancy. With the structure formed, we can now examine its primary function. The main role of the placenta is to facilitate the physiological exchange of essential molecules. It transfers oxygen and nutrients from the mother to the fetus and moves carbon dioxide and waste products in the opposite direction. However, it is crucial to understand that the placenta acts as a strict physical filter. It allows these tiny molecules to pass through, but it prevents the maternal and fetal blood from ever directly mixing. This separation is vital because direct mixing could trigger a catastrophic immune response or expose the fragile fetal cardiovascular system to the high blood pressure of the mother's circulatory system. Beyond acting as a physical filter, the placenta has another equally important role. It functions as a massive, temporary endocrine gland. Throughout the pregnancy, the placenta produces high levels of crucial hormones, most notably progesterone and estrogen. These hormones are absolutely necessary to maintain the uterine lining and support the ongoing growth of the fetus. Furthermore, these placental hormones play a critical role in modulating the mother's immune system. Because the fetus contains genetic material from the father, it is technically a foreign body. The hormones produced by the placenta help suppress local immune responses, preventing the mother's body from attacking and rejecting the developing baby. Having covered its functions, let us explore how this organ varies across species. Not all placentas are built the same way. In developmental biology, placentas are classified into different types based on the number of tissue layers that separate the maternal blood from the fetal blood. This structural classification is incredibly important because the number of layers directly dictates how efficiently substances can pass through the barrier. A placenta with many thick layers will have a slower rate of exchange, while a placenta with fewer, thinner layers will allow life-saving molecules to travel from the mother to the baby much more rapidly. Let us look at two specific examples to illustrate this difference. To see how these layers affect exchange, let us compare pigs and humans. Pigs possess what is called an epitheliochorial placenta. In this type, there are 6 distinct layers of tissue separating the maternal and fetal blood, acting as a relatively thick barrier. In stark contrast, humans have a hemochorial placenta. In the human placenta, the maternal blood actually leaves its vessels and directly bathes the fetal capillaries. This leaves only 3 incredibly thin layers for molecules to cross. This structural difference is profound. The thin hemochorial barrier in humans allows for a highly efficient, rapid transfer of oxygen and nutrients, which is necessary to support the complex development of the human brain and body. Understanding these structural differences brings us to the real-world medical implications. Proper placental function is a matter of life and death for the fetus. In human medicine, complications like preeclampsia or placental edema can cause the tissues of the placenta to swell. When this happens, the physical barrier between the mother and baby becomes abnormally thick. As we just learned, a thicker barrier makes it much harder for molecules to cross. If the placenta fails to transfer enough oxygen and nutrients, the fetus will literally begin to starve. This restriction can lead to severe developmental delays, fetal growth restriction, or in the worst cases, fetal death. To truly grasp this, we need to look at the physics of diffusion. To understand exactly how a thickened barrier harms the fetus, we turn to physics. The transfer of oxygen across the placenta relies on simple diffusion, which is perfectly described by Fick's first law of diffusion. This formula calculates the exact rate at which molecules travel through a physical barrier. Let us break down the symbols. J is the total rate of diffusion, telling us how much oxygen crosses per second. D is the diffusion coefficient, a constant for the specific molecule. A is the total surface area. A larger area means more doors for molecules to pass through. Delta C is the concentration gradient, the difference in oxygen levels between mother and baby. Finally, delta X is the thickness of the barrier. If surface area or concentration increases, the baby gets more oxygen. But if the barrier thickness, delta X, increases, the rate of diffusion drops dramatically. Now that we have our formula, let us see it in action with a healthy human placenta. We are given specific values: a diffusion coefficient of 2 × 10⁻⁵, a massive surface area of 100,000 square centimeters, a concentration difference of 0.05, and a very thin barrier thickness of 0.005 centimeters. We want to find J, the total rate of oxygen diffusion. First, we substitute all these known biological values into Fick's law. Next, we multiply the numbers in the numerator, which gives us 0.1. This represents the total driving force of oxygen. Finally, we divide this by the physical resistance, our thin barrier thickness. The result is 20 milligrams per second. This robust, high-volume supply is absolutely critical for ensuring healthy cellular respiration and rapid fetal growth. Let us compare that healthy state to a medical complication. Imagine the mother develops placental edema, causing the tissues to swell. This swelling doubles the physical thickness of the barrier, making delta X 0.010 centimeters. All other variables, the diffusion coefficient, surface area, and concentration difference, remain exactly the same. We substitute our new, larger thickness into the denominator of Fick's law. Because the mother's blood supply and placental size haven't changed, our numerator still simplifies to 0.1. However, we must now divide this constant driving force by the new, larger barrier thickness. The result is 10 milligrams per second. Because the barrier thickness doubled, the amount of oxygen reaching the fetus was cut exactly in half. This mathematically demonstrates why placental diseases cause severe fetal distress. To solidify our understanding, let us look at this schematic diagram of the placental diffusion barrier. On the left side, you see the maternal blood pool, which holds a high concentration of oxygen and nutrients. On the right side is the fetal blood, which has a lower concentration. Separating them in the middle is the physical placental barrier, representing our delta X. The large arrows passing through the barrier represent J, the actual flow of oxygen and nutrients, driven by the concentration gradient, delta C, shown at the top. As we proved with our calculations, if that central barrier becomes thicker, those arrows representing the flow of life-saving oxygen will shrink, directly threatening the fetus. This visual perfectly encapsulates the delicate balance of placental exchange. We have covered a lot of ground today, so let us review the most important ideas. First, the placenta is a remarkable, temporary life support organ that bridges the mother and the developing fetus, acting as a physical filter and a crucial endocrine gland. Second, we learned that placental structures vary, ranging from the thick epitheliochorial type in pigs to the highly efficient, thin hemochorial type in humans. Third, by applying Fick's first law of diffusion, we mathematically proved that a large surface area and a thin barrier are absolutely essential for maximizing oxygen transfer. Finally, we saw how pathological conditions that thicken this barrier can cut the oxygen supply in half, endangering the baby. You now have a complete, foundational understanding of how this miraculous organ sustains life before birth.